12th in the online classes. This is your biology subject and the chapter is sexual reproduction in the flowering plants. We are taking the topic microsporogenesis. Students, first of all we should know the structure of the anther inside the microsporogenesis takes place. So anther is bilobed and dithecous. It means each anther has two lobes and in each lobe two theca are present. Theca are what? Theca are the chambers that are present inside the each lobe. It means two thecae in each lobe. Theca are like the chambers. Anther is tetragonal structure, two microsporangia in each lobe. Look at the diagram here. It is a structure of a stamen. Upper part is anther, lower part is filament, and in between the anther and the filament there is present connective which is hidden here. So the second part is the TS of the anther. We cut the upper part of the anther in transverse section and what we are getting? Two theca in each lobes. One lobe is right side, second lobe is left side, and in each lobe two thecas are present. It means four chambers are in two lobes. Here is a line of dehiscence. This line of dehiscence indicates that when the microspores matures inside the microsporangium, this line dehisced or ruptures. So from where the microspores or matured pollen grains comes out. This is the structure of microsporangium. There are four microsporangium in the anther. That denotes the two theca in each lobe. This microsporangium has four layers. The outer one is epidermis, second is endothecium, third is middle layer, and the fourth is tapetum. These three layers are present on the four sides of the microsporangium. Let us see the function of four walls of microsporangium. The first is epidermis. It is the outer single layered protective layer. Single celled. Single cell is made up of single cell outermost layer and it is protective in nature. What they are protecting? It protects the inner three layers that are present in the microsporangium. The second is endothecium. Second, thick cells made up of thick cells, protective layer and helps in dehiscence. Dehiscence means at the time of rupture of the walls, the endothecium helps in the rupturing of the microsporangium. Third is middle layer. It is three layered, also protective in nature and also helps in rupturing of the wall. The fourth one, and is very important, that is tapetum. It was asked in the board examination that what is the meaning of tapetum and what is the function of tapetum and where it is located. So tapetum is the fourth layer. It is the innermost layer, very dense with the cytoplasm and with many nuclei. It means inside the tapetum many nuclei in the cytoplasm are present. It provides nutrition to the developing pollen grains. The pollen grains which are developing inside the microsporangium are get nutrited by this tapetum layer. So here microsporogenesis. What do you mean by microsporogenesis? It is a process of formation of microspores from microspore mother cell by meiosis is called microsporogenesis. What is meiosis? Meiosis is a process in which the diploid number of chromosome gets half by the process of meiosis and haploid number of chromosomes creates. Meiosis occurs in the sporogenous tissue of microspore mother cell. Students, inside each microsporangium there is diploid sporogenous tissue, or we can say microspore mother cell, or we can say pollen mother cell are present. So meiosis occurs in this sporogenous tissue and haploid microspores are formed inside the microsporangium. There are many microspore mother cells, not a single cell is present. There are many microspore mother cells are present inside the microsporangium, and each microspore mother cell forms 4 microspores that are arranged in a tetrahedral structure. So, one microspore mother cell produces 4 microspores. Microspores or pollen grains are formed at the end of microsporogenesis. When the process completed, then the microspores or pollen grains are formed. This is the diagram of ruptured microsporangium. There are 4 microsporangium are there, and from the line of descent, look at the diagram. The rupturing takes place and the pollen grains are coming out from the microsporangium. The outer wall is epidermis, the second is endothecium, and these connective tissues which connects the upper and lower layer with each other. Pollen grains are present inside the microsporangium. Before dehiscence, the microspores are present in a mucilaginous sheath like layer, and when it get hydrated, the pollen grains also get hydrated by the mucilaginous layer. And during the time of dehiscence, during the time of rupturing of the wall, the dehydration inside the microsporangium takes place, and the pollen grains again dehydrated and get mature and are ready to come into the environment. Students, according to our study of today, I have given you some questions for a revision. The first is, what do you mean by bilobed and dithecous? How development of microspore takes place in microsporangium? And the third is, what are the layers of microsporangium? Write their names with their functions. So follow the questions and revise it. Thank you, class.